Okay, this video is going to be done in two parts. One part is going to be me just talking about the algae experiment, which is slightly different from the earlier one with the core course. So this is the newer one for this summer 2025. And then I'm going to show you the cells under the microscope. So I'll do a second video where I'll just have the cells under the microscope. I'll focus on them. I'll talk about the cells and how you're supposed to count them during the different experiments. So in this experiment, you're going to be getting a culture of algae. When you pick up your kit from the college, you should get a little tube like this. It's going to be sitting separate. There's a test tube rack that they're sitting in. They're not in the bag. You have to get this tube and pick it up because we need to get it as much light as possible. They're live algae cells in here, green algae called Tetraselmis chiu. So you're going to get this, bring it home, and then set up your experiment. With the experiment, you're also going to have a container full of seawater, and what you'll do is you will get 3 tubes. Now, if you're in one of the sections, you're going to get a little rack like this one. If you're in the other, you're not going to get the rack. So you're going to have to put it in little containers like this. So just any kind of little bottle, cup, whatever, you can just hold your tubes in. It doesn't really matter. Okay, so you have your tubes. You're going to add a specific amount of water to the tubes, and you're going to add a specific amount of this algae. The thing is, you're going to have 15 milliliters, so 5 milliliters are going to go into each of these, and then you're going to fill it up to 15 milliliters. That means you need 10 milliliters of this to go into each of them, and you have 50, so you need 30. So you should have extra of this. This is salt water. So keep this in mind. These are marine bacteria. If you put the 5 milliliters into these containers and then just add water from your tap, you're probably going to kill them. I don't think they're going to survive at that salt concentration. So you have a container in your kit that is full of water. Right? It is salt water. So you need to use that salt water when making up the tubes for this experiment. One of them is going to be in light. The rest of the experiment's the same as the previous experiment. One of them is going to be in light. One of them is going to be in like 24 hours constant light. One of them is going to be in dark, 24 hours constant dark. And the other one, you're going to have to just put in a normal light-dark cycle. And the idea is you want to see over a period of time how the cell number changes. But keep this in mind, before you begin the experiment, before you put it in the tubes and do the, you need to have a concentration. So technically they'll all be the same when you start off. So you're going to take a little bit of this, put it in there, and then you're going to read a sample. So what I'm going to do is show you that little part. You should have in your kit a little container like this. Inside it is a slide. It's a counting slide. And the slide already has a little cover slip attached to the surface. There is a top and bottom. There's an A and B on it. Make sure that when you're looking at it, they're not, well, the A you wouldn't see as backwards, but the B you would see as backwards. Also, if you run your finger across it and it's perfectly smooth, that's the bottom of this. You need to turn it around. If you run your finger over, you're like, oh, there are little grooves in there. There are little openings. That's where the liquid will go in. So what you'll be doing is, after you do the dilution, so you'll take some of this, put it in the tube, all 3 tubes. Add your water to the tube, so now they're all exactly the same. That's when you want to count them, before you begin the experiment. Not from here. I don't have the salt water here, so I can't do this to demonstrate, but if this were one of your already diluted tubes, all you need to do is with your little eyedropper, you need like 1 drop. You don't need any huge amount. You just take 1 little drop, and there's a little half circle area here. You're just going to put that drop to the circle, and you'll see liquid get sucked right under the cover slip. That's it. You didn't really use anything much of it. You need a very, very tiny amount. Something else to keep in mind. If you look at your tubes over time, you may notice it's very green on the bottom and a lighter green throughout. Some of the cells are motile, so they will be swimming around, but you should invert it like this with the cap screwed on tight to kind of mix them. When the tubes are just sitting and you're not doing anything with them, you should loosen the caps so they're loose. Okay, so the cap shouldn't be tightened when it's just sitting there during the experiment. But if you're going to take a sample from the tube, don't forget that the cap is loose. Make sure you tighten the cap, then go ahead and mix it, and then you can undo it and then go ahead and take your sample. So now I have a sample on my slide and I want to look at it. So you need your microscope that came with the kit. You're going to need you're going to need a phone camera to take photo and videos of this, partly because it's going to make it, one, very easy to count the cells with it. Without it, it's actually pretty difficult. Secondly, you have to show proof that you actually did this. If you turn in the data and you answer a bunch of questions, but I don't see a photograph of your slide set up on your microscope, and I don't see a photograph taken through the microscope of the cells on the slide, you can't get credit for doing this because I don't know that you actually did it. So you have to actually show me that you did it with those pictures. So I'm going to go ahead and get my camera, attach it on here. I'm going to take the slide. Remember, there's a little in this microscope base. Now, these bases are unique. We handmade these for the classes to go along with these microscopes. The scopes work okay without the base, but the idea is in microscopy is this mirror underneath here will change the angle of light, and the angle will change the contrast of what you're viewing through the microscope. So when this microscope shines the light down, it's going to hit the surface of the slide, and that's all you're going to see is the light coming from the top. But with this particular base, when you put the microscope on it, there's a mirror in here. And then with this little knob, you're going to be able to turn that mirror and adjust the angle of the light that's coming back up at the slide, and you're going to get really good contrast. You're going to see the little grid. There's a grid etched onto the slide. You'll see the cells. So you're going to need to be able to adjust that. So we put these little knobs on here. It's very tiny, so it's really tricky to use at first, but you should get the hang of it after the first couple times of using it. Your first experiment is just with yeast cells, and that's almost intended for you just to get used to using the scope and using the base and counting some cells. Those cells don't move at all. They're just sitting there. So it's a lot easier to do that one. This one with the algae is like the next step, the next level up. So you need to have a little bit better skills to be able to do this one. All right, so what I'm going to do now is I'm going to get the camera, I'm going to attach it here, and I'm going to take some pictures of these actual live cells, the same ones you're going to see, so you know what do the cells look like. If you're looking at the cells, you're not looking at the cells. Okay. So that's the end of part one, just the introduction to this. Part two, you're just going to be seeing through the microscope.